So this is where we left off in the lesson on today, or today I'm making the video. We're probably watching this on Tuesday. And so the relationship between cyclin and CDKs was found out by these three scientists down here. And what they noticed is that when cyclin peaked in its amount in the cell, that the cell was going under mitosis. And they noticed that there was this chemical called cyclin, that's why they called it cyclin, that was cycling at levels through the cell, and it was high when the cell was undergoing mitosis. They began to call the Cyclin CDK association the MPF, or the mitosis promoting factor. So when cyclin is bound to CDK, they gave it a name called the mitosis promoting factor. Now, there are lots of different cyclins and lots of different CDK molecules that work at each of the checkpoints. We're now just talking about the G1S checkpoint. But again, they noticed that the level of cyclin was high when the cell was going under mitosis, and they called the cyclin CDK molecule, that when they're associated together, they called it the MPF, or the mitosis promoting factor. So you might think that once cyclin is there, well, if cyclin's there, it's going to be dividing forever. But there are times we're going to want the cell to stop dividing. And so we have to get rid of cyclin. And how do we get rid of cyclin when we're done? And the answer is that we ubiquitinate it, or we add a protein tag, which is known as ubiquitin. And when we add ubiquitin to a molecule, we call it ubiquitination. And that signals for it to be degraded. Okay? And there's a new organelle that we're going to learn. The new organelle, we didn't learn first, called the proteasome. And what happens is, let's say this is cyclin, our protein molecule cyclin. We add ubiquitin to it, which we usually abbreviate UBI, and that signals for the proteasome to come and degrade it. Okay, now, this usually happens when the cells are out around the cell that are producing growth factor, stop producing growth factor. And this is not fully understood, but that signals for the cell once the ubiquitin, and we'll get into this a little bit later, once growth factor is no longer being produced, and the cell is no longer experiencing it, it ubiquitinates cyclin, and the proteasome comes and degrades cyclin, so therefore it can no longer bond to CDK, and then the DNA replication proteins will no longer be activated. But again, the point is, how do we get rid of cyclin? We ubiquitinate it, which is adding a protein tag called ubiquitin. And that signals for a new organelle called the proteasome to come and degrade it. So we answered this question already back on the previous slides. We asked what causes cyclin to be produced. And we've talked about this growth factor is produced by cells in the environment, which causes the cells that respond to the growth factor to produce cyclin. Now, if we go back to the previously, I wrote down what I wrote on the board. This is the part that we don't know yet, or we don't understand. So yes, we know that growth factor produces cyclin, but this arrow is a big sort of empty box. How does growth factor produce cyclin? And that's what we're going to learn next week, is how does growth factor make cells produce cyclin? That's what we're going to learn next week, but that's sort of like a big empty box right now, is sort of how does growth factor, which is outside the cell, get the cells around it to produce cyclin, and that is a big empty box that we're going to fill in next week. So at the beginning of this lesson, we talked about the fact that there are go signals and there are stop signals, and we've learned the go signal. So we have growth factor, which is abbreviated GF a lot of the time, which causes the production of cyclin, and I'm writing with the mouse, so it's going to be awful. And then that binds to CDK, and then we have CDK, and then CDK cyclin complex, and you can probably write this better than I can with the mouse. So the CDK cyclin complex will push the cell cycle forward. Okay, but there are also stop signals. If you remember, and I'm not going to write it because it's going to take too long, there were two things that would stop the cell. If it wasn't big enough, right? Or if it wasn't attached to that thing called the ECM. You remember the ECM? If that's the case, okay, and we're not going to go into the details of this, what happens if the cell isn't big enough, or it's not attached to the ECM, there are proteins that sense this, and then they inhibit cyclin from binding to CDK. So even though the growth factor is there are proteins that are produced when the cell's not big enough, or when the cell's not attached to the ECM, that inhibit cyclin from binding to CDK, even though it's there, and then the cell cycle can't go any forward. So that's sort of like the break on this go part of the checkpoint. All right, answer the guided questions, and I'll see you Wednesday after the PSATs.